Dear students, welcome to our last lecture on bioinformatics in this course Applied Environmental Microbiology. Today we will be continuing the conversation on an actual tool that is available online anywhere in the globe to anybody who wants to use it to analyze the big data that we generate or to analyze the data we generate from our sequencing, and this is very essential for an applied environmental microbiology engineer and student to know. So last class we talked about NCBI, which has a very important function called BLAST, where it does a very basic local alignment and it searches the most similar sequences or proteins for the entry that we have put. It has various options and I briefly introduced you to them. I also mentioned to you that sometimes BLAST P or BLAST N is better than BLAST X, sometimes the other one is better than the other. It is very important for you to first of all experiment and try all of them when you generate your sequence, and then try to understand if your sequence is of a region that is very well established, very well curated, like 16S rRNA, then a BLAST N would be sufficient, do not need to go to BLAST P. Also in 16S rRNA, it is the structure of 16S ribosomal unit that is more important. But if you are looking for a functional gene, for example CEL48, which is associated with glycoside hydrolase activity during cellulose degradation, in that case you might consider doing BLAST P instead of BLAST X or BLAST N or BLAST X, not BLAST P but BLAST X, because then it will translate your nucleotides into protein sequences and then blast your protein sequences with a protein database. And that is important because in environment the cellulose degrading enzymes are highly degenerate, which means if there are 3 different codons that can code for one amino acid, then probably in some microbes there will be one particular codon used, in some another codon, and in the some third one. So if we do nucleotide search, we might say, well, the gene is not similar because in our database we only have codon 1, not the other 2. But if you translate it into protein and you do the similarity of the amino acids, you will see a lot of similarities. Alrighty, there are some other options also available to you when it comes to aligning your sequences that you generate from Sanger sequencing or the small number of sequences that you generate from anywhere. The most popular among them, if you are interested in typing it, if you are interested in annotating your sequence, and you want to compare, and this is when you are comparing the ribosomal unit, whether it is 16S in bacteria or 18S in eukaryotes and 18S in archaea. Remember, 16S in bacteria and 18S in archaea and fungus in eukaryotes. So regardless of what it is, we need to the best option that we have is RDP, which is Ribosomal Database Project. So you can just Google Ribosomal Database Project, it is maintained by MSU, and you can go to sequence match. So when you go to sequence match, make sure that you keep your sequence ready here. So here we have sequence ready, and let this open. So once you open the sequence match, it will ask you to log in the RDP, but if you, and I highly recommend that you have an account in RDP, and if you do not, just log in as a guest. But with the guest, the problem would be that your data would be not stored. If you log in as your user, then they will store your data and then you can access it over and over again. But as a guest, your data will be deleted, much like what happens in NCBI. Okay, anyway, so RDP is one of the options, and when the page uploads I will tell you more about it. Meanwhile, the other options that we have is Green Genes. So if you can download data from database from Green Genes, and this is, by the way, greengenes.lbl.gov.download. So I have everything ready here. So you look at Green Genes here, and this is your Green Genes, and the good news about Green Genes is that this is a very well maintained database. It is highly curated. So if you get a good match with Green Genes, we can be very sure that whatever match we are getting and whatever prediction it is making is probably true. The next option you have is Silva. Silva is also very well curated, but not as highly limited as Green Genes. So with Green Genes, the probability of getting a good match is very low, but with Silva the probability is pretty nice. You can also download this database and then use bash commands or use different platforms, and the information is very well documented under the documentation center, where you can find different kinds of tutorials. The other thing that Silva will do is it offers you different kinds of tools, and you can go through them very quickly. It will allow you to allow you to download the database. It also has an Silva NGS, where you can use your high throughput sequencing data and then with your high throughput sequencing data, you can use Silva NGS to make sense out of it. We also have Silva tree viewer. So many a times many programs will allow you to make a tree, but you can't view it. So remember, the tree here is your phylogenetic tree, how similar the sequences or samples are to each other. So they will give you a matrix which will have all the data you need to actually draw a tree, a dendrogram, cluster dendrogram, but it will not make one. So if you have Silva tree viewer, you can input your file and then it will make a tree for you. Then we have ARB, which is a big project, and then here they have UniEWEC, which is Universal Taxonomic Framework for Eukaryotes. And then another very important part of Silva is the SINA Online. So SINA Online is very much like NCBI. So if you have few sequences like 1, 2, 3, 4, 5, 6 sequences, not a lot, you can do SINA alignment. So you paste your FASTA file here. So we have pasted a FASTA file here, right? Perfect. And you can also upload a file if you have if you have a file properly maintained, and then you can choose different options. This is SSU gene, yes, we want it attached to the last align base, or you want it attached to the last line, just leave it at last line base. And then you can also look at the advanced alignment parameters. We are expecting it to be bacteria, so but anyway we can just write none. Alrighty, and you can give it a name if you want. Query 1. Very good. And then these are different parameters, you can change them as per your requirement. And if you click search and classify, what it will do is it will not only search the similarities to all other Silva database entries, but it will also classify your sequence for you. It will tell you whether it is gamma protobacteria or not. But remember, the first one according to NCBI was gamma protobacteria and second was Gematria monadellis. Let's look at what this is. So let's run aligner. Alrighty, so our query 1 is under queue. So when this query 1 will be done, we can actually open it and we can take a look, we can show the result, we can download the file, and we can add our neighbors to the cart. Anyway, so SINA is a very good tool available from Silva for amplicon sequencing, and this is Green Genes. I highly recommend both of them. And let's see what RDB, okay. So in RDP this is not opening properly, but you can cut and copy paste your sequences here. You can tell them what kind it is. This is uncultured, the size is less than 1200 base pair, quality is good, we are interested in nomenclature, and then you can have different kinds of options, and then you can submit it, and then it will also give you data. Okay. And then this is the translation tool, by the way, just to remind you, XPASI is the most popular one. Now let's come to high throughput sequencing. So in high throughput sequencing, what happens is we generate lot of data, tremendous amount of data, and so we have thousands or millions of sequences, and there is no way that we can use an online tool like NCBI or like SINA or like RDP to classify our sequences. Another thing is with high throughput amplicon sequencing, when we are talking about amplicon sequencing, not the whole genome sequencing. So if you're not clear about what's the difference between two, I highly recommend go back to previous lectures, figure it out, and then watch this lecture ahead. Alrighty, so if you are watching it now, you know the difference between high throughput amplicon sequencing and whole genome sequencing. So in high throughput amplicon sequencing, in this process we have a lot of errors, we have a lot of chimeras that may come up, so we require a dedicated tool to analyze it for us and then give us the data, the meaningful data. MGRAST is an online platform which is very popularly used. It's almost the industry standard right now, and it's a metagenomics analysis server, that's what they call themselves. And what you can do is you can upload your data, you can allow it to analyze, clean your data, remove the chimera, remove the errors, then you can download the results, and then you can analyze it here too. So one of the first things you need to do is you need to log in, and in order to log in you need to give them requests, and then they take few days to get back to you. So please don't leave anything for last minute, because MGRAST, remember we are dealing with high throughput amplicon sequences, so we have lot of sequences, lot of data. It will take anywhere from few days to weeks for the MGRAST to finish its processing of your data. And MGRAS gives you multiple options. You can choose to make your data public upon completion of analysis by MGRAS. In that case, they will give you priority because they promote transparency and free sharing of data. Or you can say that I want to make my data public after 6 months or after this much time, then you will be next in order on priority list. And then you can say I do not want to make my data public at all until I choose otherwise, then you will be on the least priority. So, you can choose what level of privacy you want for your data, and then your priority on the queue will also be a factor. Alright, let us look at some examples. So, this is mine MGRAS, and what I want to do is I want to see what is going on with my data. So, I can click on my data and it will bring my data, but let me warn you, MGRAS tends to be slow because it is dealing with very heavy data and a lot of data. So, the server tends to be little slow. So, please do not, if you have amplicon sequencing data, do not leave your analyses for last minute. So, you can click on nanocellulose here, and this is my project, by the way, where I have clicked nanocellulose project, and then we will wait for it to come up. And then these are the sequences that I had submitted. Note, these sequences are not high throughput amplicon sequencing sequences, these sequences are whole genome sequences. So, MGRAS is for whole genome, and for high throughput amplicon sequencing, the two most popular are Mother and CHIME. We will be very briefly talking about Mother and CHIME. Alrighty, so what I can do is I can pick one of these sequences. For example, let us say I am interested in AR12R1, and this is a sequence that I had submitted, and MGRAS took a week or so to analyze the sequence, and then let us see what kind of output data MGRAS will give us. So, this is going to take a bit to open up. Meanwhile, what I will do is I will open up for CHIME and Mother. Alrighty, another thing that I can do is I can look at, now here I have different options in MGRAS. This is home, takes me back to the original page, okay, and then this is search, where I can search different things. This is upload data, if I want to upload my data. This is download my data, freely available data, and this is analysis. So, I have clicked on analysis. To click on analysis, it will give me a long list of what I want to analyze, and I can choose. So, let us say I want to analyze my own data. Let us say I want to analyze A12.R1. So, I want to type here okay, let us see if this is there. Alrighty, so I have one file called A1R2. I can select this file. The other file, let us write I want to look for nanocellulose, because I know some of my files are generically referred to as nanocellulose. Alrighty, so I will take this one, and I have accepted 2 files. So, now what I can ask the MGRAS is to do, yes, I want to compare these 2 data. So, these 2 samples. Now, this is going to take some time. So, let us move on here, no progress yet. So, let us see what is up with our CINA. Again, this is going to take time. So, let us say I want to look at this particular file. So, we will open this file. And here look, it is actually been telling me how much of data it has processed so far, how much it has not processed. One thing you want to note as you are waiting for MGRAS to open up the data, because as I mentioned the data is huge, so it takes time, is the kind of information they require from you when you are submitting your data. They need to know what name you want to use, and then obviously they can calculate the base pair count, the sequence count by themselves. Then you need to inform them what kind of sample it is, where did you collect the sample, from which country, what are the coordinates, what kind of sequencing you did, and what is the method of sequencing. So this metadata they require because, as I mentioned, they promote free sharing of data. So if I just have the file here without knowing where it was collected from, when it was collected, what kind of sample it is, and what kind of analysis was done, what kind of sequencing was done, then I cannot use the data for my own analysis. So this metadata is very, very important for us. Without metadata, it will not accept your sample. You need to submit metadata. So for now, I looked up the sample A1 to R1 and I wanted to know what are the qualities of this sample. So I clicked on it. It took a bit for the web page to upload, but this is the kind of information it will give you post analysis of your sample. So it knows that I chose privacy. I chose that I don't want to share this information because I was not sure when I'm going to publish this. And then it tells who was the lead PI of this research. And then look here, it will give more generic information about the sample. The data set was uploaded on this time at this date. It has these many sequences, these many base pair. The average length is 108 base pair. And then it will give us a basic summary of what percentage of sequences did not pass the quality control pipeline, how many had dereplication, and the ones that passed, how many had RNA, ribosomal RNA sequences, how many had sequences that are that we know are associated with proteins with predicted functions, and then how many had predicted proteins that we do not know the function of, and this basic summary it gives. And then it draws some graphs that are very helpful. Same information but written in graph form. So it will give me a sequence breakdown. It will tell me that what percentage, 10.72% failed quality analysis, quality control, 6.88% is unknown. This is important. These sequences could be chimera because they are not known, they are not present in the database. It is also possible that these are some different kinds of genes that we have no idea of yet. And these are the ones, 82.4% that we predicted, that we know. We have information in database of. The second one tells you about predicted functions. So majority of the predicted proteins were annotated, so we know what their function is. But 33%, 1/3 of them, we do not know what their function is. A very tiny portion of it is ribosome. So if you want to find out what kind of microbes are present in your sample, going for whole sample metagenomics may not be very good idea, because you will be sequencing lot other more proteins, lot other more DNA than just one that stands for 16SR RNA gene. In that case, it would be better for you to go for high throughput amplicon sequencing and focus just on this narrow strip of ribosomal RNA, which is less than 1%. Then here it will give you basic analysis statistics, like how many base pairs were uploaded, how many sequences were uploaded, and so on and so forth. And then it does the DRISI analysis. So this DRISI analysis will tell me about duplicate reads and where do I lie in the distribution of duplicate reads. And then this is Kamer profile. Kamer profile basically will tell how many times the abundant sequences appeared, and then from most abundance we fall to less abundant. And then this is nucleotide histogram. Nucleotide histogram gives a distinguishes between the different kinds of nucleotides that are present in our sample. For example, A, T, C, G, and N, empty, nothing predicted. And we can get an idea. This is very important for us to know because we do not know what it is. It's ambiguous. Okay, and this is very important because GC ratio gives us an idea of what kind of microbes are there. And then source hits distribution. So after the quality checks were done on the sequences that I uploaded, it matches them with different databases, and here are the databases. And when it does that, it's trying to give me an idea of how many reads were annotated. So if the E value is very low, it is in red. If E value is very high, it is in blue. And we notice that most of the reads happened were in subsystems, RefSeq, and in Patrick, and not so many in SwissProt, quite some in GenBank, not so many in KEGG. So these are all different tools, different databases that are helpful for us in different perspectives. So GenBank definitely for genes, KEGG for metabolic pathways, and so on and so forth. RDP is a ribosomal database project that we talked about, very important. Anyway. So next, it also tells me about functional category hits distribution. So it will tell me already. So now this it will tell me for COGS, KOs, and NOGs, and subsystems at highest level of functional hierarchies. So we know that 42%, nearly 43% were involved with metabolism, and some 16% were not characterized properly. Orange were for information storage and processing, the red were cellulose processes and signaling. So metabolism, unless I am interested in certain functional traits related to metabolism, this is housekeeping. This is housekeeping. This is also mostly housekeeping, and this we do not know. Alrighty, when we look at NOG, similar information. This is more subsystem, we have more detailed information. So most are clustering based subsystems, and we have carbohydrates. This is what my research was interested in, by the way. Then we have amino acids, protein, and so on and so forth. Then it will also inform me about taxonomic hits. Most of the sequences were bacterial samples, nearly 99%. Very small portion of them were archaea, eukarya, archaea, viruses, and others. So if I go for on phylum level, I see that most of them were proteobacteria, some were bacteroidetes, some were firmicutes. I was very interested in bacteroidetes and firmicutes, and then actinobacteria, chloroflexi, and so on and so forth. And then on class level, most of them were beta proteobacteria, followed by bacteroidea, followed by gamma proteobacteria, followed by clostridia. I was very interested in clostridia and in bacteroidea. Then in order level, again it divides into different parts. Again, Clostridialis was very important because this was a cellulose degrading study. And then it will also tell me on family level. Now look at these plots, it's very hard to tell what the data is just by looking at these pie charts, but you can actually download the data here, and once you download the data, you can actually draw your own diagrams and do your own analyses. So this is the beauty of MGRAS, it analyzes things for you, and this is on genus level. Alrighty, and then this is rank abundance plot. So this is the abundance of the taxon, and this is which taxon we are talking about. Lot of Bacteroides, least amount of Verminopherobacter. And then this is rarefaction curve. So if you look at rarefaction curve is also called collector's curve. What it tells me is that was my sampling depth sufficient or not. So if you look at my rarefaction curve, it has not plateaued, which means that the number of new species that we could have detected was, if we had more sequences, if we had more sequencing depth, would be higher. So we have not, this data is not completely representative of all microbial members in the community. So this is very important curve, and this is very important information. So if I have like this, then I have to accept the limitation in whatever conclusions that I make, that this data is perhaps not very good representation of what's really happening, because the collector's curve or the rarefaction curve has not plateaued. So once it starts plateauing, once it plateaus, we do not detect any more species, no matter how much you increase the sequencing depth. So you have reached the optimal sequencing depth. Then we have alpha diversity, and then we also have sequence length dentagrams. Most of them were around 108, so this is good. This is the target length of sequences. And then also tells me GC distribution. It gives me more information about sample, people who had submitted the sample and so forth. So for each sample you will get this information, and for basic analysis this is enough. Alrighty, it is also possible that we can compare two samples, and in this is where the beauty appears. So in MGRAST, once you have uploaded your data, and it might take some time, just the mere act of uploading the data, you got to either decide to make it publicly available or make the data private. Now for example, the data that I was uploading in this lecture earlier is private data. So now the issue with private data is that if your data is private, obviously the benefit is that no one can view it and use your data and publish results. But the issue with that is that the algorithm, the processing by MGRAST takes long time, because it is given the least priority. Whereas if you make your data public, you are likely to receive data analyses in maybe a couple of days, or maybe maximum couple of weeks. So here I want to give you a glimpse of how MGRAST results may look like. In previous examples I showed you per sample analysis of the metagenome that you have uploaded on MGRAST. Here let's take a look about how we can use the data from MGRAST to make comparative analyses between different samples. So here on the left panel here, we have this particular dendrogram from one sample, and we note this is from MGRAST, by the way, and you notice that MGRAST can give you information on class level, phylum level, or family level, species level. You decide the level
of resolution that you want your data to be represented. And these dendrograms are very helpful for us to understand what's the diversity of your sample and how many different kinds of microbes are present, and this helps us also understand what kind of functions might be happening, because for example, if Clostridia is present in large quantities, we can suspect that maybe this is cellulose degrading environment, or if delta proteobacteria are present in large numbers, we can suspect sulfate reducing environment. Another thing about MG-RAS results is that not only does it give you a dendrogram that informs you about which of the microbes are present, but it also gives you an idea of in how much abundance. So if you can find out these little bars here in your data, they will give you idea of relative abundance of the sequences that matched a particular species, class or family or phylum. Now this information can be used to generate another kinds of plot, which is on the right panel here, where you see not only can you see the different kinds of microbes present, which species, which classes are present here, but now you can make comparative analyses. For example, there are two different kinds of samples being analyzed here. One is from a healthy mammal and the other is from diseased mammal. And we can compare which microbes are present more in healthy animal versus in diseased animal. And this is a very good visual image that has been made using data that can be made using data from MG-RAS. So for example, we can clearly see that Streptococcus was present in high number in healthy mammal, whereas Treponema and Prevotella were more present in unhealthy mammal. And not only that, you can also get the information on what's the overall abundance, which is given by the blue circle. It's actually not a circle, but it is a layout of blue bars. And this is one example of how you can use MG-RAS data to make really good visual representations of your data. Now, there are many packages on software R, which I have recommended earlier in this class, and I highly recommend you to explore these packages that you can use to draw wonderful diagrams and make your data more clear and more accessible for scientific and general public. Now, here is the data that I had uploaded in this lecture earlier. Here is some analyses from that data. So, not only can you actually download the figures made by MG-RAS itself, for example, the one on the left, but what you can do is you can download data at every step of processing analyses, and then you can make your own graphs to make your own analyses. So, these are 8 different samples that were uploaded on MG-RAS and analyzed for my nanocellulose project. And I was very interested in glycoside hydrolase family of enzymes, and now here I downloaded the data and found the enzymes that are that belong to glycoside hydrolase family and have similar functions, the functions that I'm interested in, namely degradation of beta glucoside bond. So now when I collected a number of sequences in each of these in these families, I could draw these bar plots that help me understand, that help me make comparative analyses from different samples. For example, this sample has been incubated for more than 200 days. It was taken from a wet land. It's the original sample, as in original for the batch assay. And then it was exposed to microcrystalline cellulose, cationic nanocellulose, and sulfuric acid reduced anionic nanocellulose. And I can see how after a period of time, which is 4 weeks, how much the glycoside hydrolase have changed from the initial inocula over time. Now similarly, this graph here is also very informative. So the beauty of MG-RAS is that if you find it challenging to analyze data or make visual representations of data, there are sufficient tools available on the online platform itself for you to make very pretty diagrams, meaningful diagrams, and you also have the flexibility to download your data and interpret it and represent it in whatever way suits you and your paper.
Let's switch back to SINA. So remember, we used Silva's SINA to align our two 16S rRNA Sanger sequences. It has finished them, and it tells me that the first one they found 95% similarity, and the second they found 91% similarity. So let's display their classification. The first one, it said, is gamma proteobacteria, but it didn't stop there. It goes even ahead and says what kind of gamma proteobacteria we are talking about here. It says it is Xanthomonadellis, and the second one is Gematimonadacea. Cool. And I also have the identity, and I can actually export it to CSV file, and then I can get more further data that I need. So students, this is SINA for you. Now when it comes to high throughput amplicon sequencing, we do not need to go to MGRAST. We can use we use other platforms, and the two leading platforms, as I mentioned earlier, are CHIME and Mother. So let's start with Mother. Mother was the one that was developed and popularized earlier. So Mother is initiated by Dr. Patrick Schloss in Department of Microbiology and Immunology in the University of Michigan, and earlier versions of Mother were called Daughters and Sons. And the beauty of Mother is that it's a very simple setup, and it does not require a lot of space in your computer to run. So it is kinder on your computational abilities, and the good news is that it is very well documented. So this is the wiki page of Mother, and if you are interested, you can look at how to download Mother, how to join its mailing list, and so forth and so on. And you also can have manuals. So let's look at manuals. So in manuals, what you can do is you can go through different kinds of operations that you are interested in, general operations, sequence processing operation, and so on, and find out what the commands are. So let's look at one particular operation that is very commonly used. Mother 16S analyses wiki. Okay. So this is the MySeq SOP for Mother. So in this, what they have, obviously they have the citation here, and now if you go through this, they'll give you step by step information on how to use Mother. This is the data, the mock data that they're using, and these are the files in the mock data. So these are comparisons. For example, this perhaps is the one end of the pair, and that's the other end of the pair, and so these are the paired readings. So when you attach them, we get one long sequence. So you make one stability file that will call your FASTQ files, and then the next step would be you want to remove the PCR errors. So they are removing the PCR errors here, and then they process improved sequences. So you want to find out what are the unique sequences, you want to remove other sequences. If you want to count sequences, count .6, and then summary dot sequences, summary dot anything, you can always get a summary, and then you can rename the file, and so on and so forth. Then alignment option comes here, and when you align, you have the option to choose the database you want to choose. Here they are using Silva, but if you want to use another database, you can use that. And then here is the summary, and then you can filter your sequences, and you again find unique. Then you can cluster your sequences. So for clustering, you do pre-clustering, then you look for chimera, and then you remove the chimera sequences that you got from chimera.vsearch, and then you can classify your sequences. You can remove certain lineages that you know should not be present in your sample anyway, and then finally you will get your final data, in which you can find out what is your error rate in this particular case, and then you can analyze. You can find the distances, so distance matrix like Euclidean distance, Break-Withers distance. You can do hierarchical clustering, which is again they have given the commands for that here. Alright, another thing you need to know, Mother and CHIME, they don't have graphical user interface. So you don't have to click on a window and then there'll be a drop down list, and then you can click on what you wanted to do, but you'll have to write the commands. Thankfully, both Mother and CHIME have very well documentation, so you will never be lost. And there's almost all kind of data analysis you need to do, they'll inform you. Alright, so we have done clustering, we can do classification, and then you can do phylotype analyses, you can classify. You can also do phylogenetic analyses, and see, this entire wiki page will help you a lot in trying to do even calculating alpha diversity, beta diversity, and so on and so forth. So this is MySeq. And if there are questions that you face when you're not able to, when you're trying to follow a particular SOP from wiki of Mother, then there is this wonderful forum here, which supports the Mother software package. So you have a lot of questions, and you can question if there's a feature you want to see, you can feature that in here. And this is a very supportive community, and very quickly someone will respond and help you with your analyses. Alrighty students, this is Mother for you. The next we have is CHIME. So in CHIME, it is quantitative insight into microbial ecology. In CHIME, now they are moving to CHIME 2, which is good. But with CHIME, it requires a platform. The computational needs are much higher than that of Mother. And in CHIME, if you're using Windows or Mac OS or Linux, you might want to install the VirtualBox, and that's the easiest way. Or you can use VirtualBox to install CHIME, or you have even installation of CHIME is a challenge actually. Or you'll have to go through, make sure all dependencies are present, and then you can install it and then run it. Similarly, like Mother, they have very good script documentation that will help you do your analyses. And again, this will do analyses for all platforms, so that's pyrosequencing, Illumina based sequencing, or ion torrent based sequencing, or applied biosystem based sequencing, whatever kind of sequencing. There will be lot. These are all commands that you can use. One difference that I have noted between CHIME and Mother is, while Mother is computationally kinder than CHIME, CHIME tends to make prettier plots because data visualization is so important. The other thing is that certain funding agencies now require people to submit their data to CHIME and to use CHIME in US. So in that case, CHIME does have an upper hand, but Mother offers lot of flexibility. So if you know coding, if you're a hardcore bioinformatician, I see that there are advantages and disadvantages for both Mother and CHIME. Both are equally good and both are equally useful. Dear students, I hope that this lecture and the previous lecture gave you valuable real information on how you can use tools that are easily available to you to analyze your sequences and make sense out of them. And with this, we conclude our lecture series on our course, Applied Environmental Microbiology. All the best for all your assignments and home exams. Thank you.